Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and we're kind of changing it up a little bit. We've been talking about meiosis and how to make gametes and what fertilization is. Today we're moving into some DNA technology, and I wanted to talk about something very, very important. It's called DNA fingerprinting. So have you ever noticed how sometimes if a criminal leaves DNA at a crime scene, police and investigators can kind of determine who the suspect is by analyzing that DNA? This is how they do that. It's called DNA fingerprinting. I'm going to run you through how to do it real quick, and then we'll probably get some practice on it online. So what is DNA fingerprinting? Well, this is a method. It's commonly used in solving criminal cases. We can also use it to determine parentage, which means we can basically find out who the dad is, okay? It involves a technique called gel electrophoresis. To do this, first a DNA sample is collected, and because the available sample may be small, a process called PCR, or polymerase chain reaction, is used to amplify the DNA, which increases the amount of available for analysis. So sometimes in that criminal investigation, a criminal or the suspect might leave behind just a little bit of DNA. What we'll do is we'll take that DNA, run it through a polymerase chain reaction, PCR, will make extra copies of it, and then we can do something called gel electrophoresis when we can basically analyze it and compare it to other people's DNA. That'll help us determine who the father of a child may be, or also it'll help us find that suspect in a criminal case. So let's talk about how to do this. So in order to make a DNA fingerprint, we have to do something called gel electrophoresis. So I'm going to kind of walk you through that real quick. Okay, so in gel electrophoresis, first the DNA sample is cut into fragments using restriction enzymes. Okay, these enzymes, the restriction enzymes, they kind of act like molecular scissors, so they can cut DNA at very specific sequences. This creates smaller fragments of varying lengths that can be separated and analyzed. What we're looking at here, a great example of a restriction enzyme, is EcoR1. It's an enzyme inside of E. coli. They use it for their own purposes, but we can use it to cut DNA into these small fragments. Cutting it into small fragments is going to make it easier to analyze. So that's the first step. Here's what we're going to do next through this gel electrophoresis. Okay, the gel part of this is an Argos gel. It's kind of got like a gelatin feel to it. And inside that gelatin, there's all of these small little holes. Okay, the gel has a consistency of gelatin, and at the top, there's these really small spaces known as wells. Okay, the DNA fragments are placed in the wells, and a dye, you'll usually add a dye to make it more visible. So again, just to kind of orient you a little bit so you know what's going on here, this will be a sheet of Argos gel. It's like jelly. At the top up here are these really small holes. They're called wells. What we'll do is we'll take DNA and put it in those wells. Okay? I'll go through all the steps here in just a second, but as an overview, basically what we're going to do, we'll run a current through the gel, an electrical current, and that makes the DNA start to spread out across this Argos gel. And what we'll make at the end of it is something called a DNA fingerprint, where all of the DNA in this first column belongs to one individual, all the DNA in this column belongs to another individual, and then we can compare them to see how similar or different they are to each other. Okay, so let's kind of go through those steps of gel electrophoresis. First, we make the gel and we put the DNA in the wells at the top. What do we do next? Okay, so after that, the gel is going to be placed in a buffer solution. Basically, there's like a special type of liquid that we're going to put this inside of. Then we're going to run an electrical current through the gel. Okay, the wells at the top are on the negative end, okay, and at the positive end, this is where the DNA is going to move to. DNA has a negative charge, and as we know, opposites attract. So the DNA is up here at the top. As the electrical current runs through it, it's going to draw the DNA to the positive end because DNA has a negative charge. So that's going to cause the DNA to spread out throughout this Argos gel. Okay? The smaller the fragment, the faster and farther it moves through the gel. The banding pattern that results is known as a DNA fingerprint. Okay, so again, what we've done here, these are 3 individuals their DNA, and we're comparing them by making this DNA fingerprint. We've run the electrical current through this gel, and it makes the DNA spread out. The short, small DNA fragments move further, and the longer DNA fragments don't move as far. So these would be like really, really short down at the bottom, and these are really long segments of DNA. We could then kind of compare and contrast them to each other, okay? So what I wanted to do next is I have some review questions I wanted to go over with y'all. So teacher, if you could, go ahead, pass that worksheet out. Y'all take 5 or 10 minutes, and then when you're ready for the review, come back to me, and we'll kind of walk through it together in case anyone was confused. All right, hopefully that didn't give y'all any problem, but just in case it did, I'm gonna kind of walk through it with y'all. Again, what we're doing here, these are DNA fingerprints, and we can use them to compare and contrast and find out who the father of a child is, or find the criminal in the suspect in a criminal investigation who likely did the crime, okay? So let's read over these and see what's going on. So the directions here say, use the DNA fingerprints below to answer the questions that follow, okay? So you are a crime scene investigator, and you run this gel electrophoresis, and you make the following DNA fingerprint. There's 4 suspects, and it's your job to find who the most likely suspect is. Okay, so at the crime scene, there was some DNA. We ran the DNA through the gel electrophoresis machine, and this is the DNA fingerprint that we got. Okay, this is the DNA of the victim. Okay, and then the police have found 4 suspects who might have done the crime. Maybe they were around the area when it happened. It's our job to figure out who of these 4 individuals most likely did the crime. We can do that by comparing the DNA fingerprints. Basically, what we want to do here is find which of these suspects most matches the DNA at the crime scene. Okay, so let's see what we came up with. Number 1 says, based on the DNA fingerprint above, who is the most likely suspect? Okay, well, let's kind of compare these. Okay, again, it can't be the victim, so I'm just going to kind of cross that one out so we don't get confused. It's not going to be the victim here. Okay, so suspect 1, they have 2 bands that are the same. Okay, so there's a couple matches there. Let's look at suspect 2. They do not have anything in common with the DNA at the crime scene. Let's look at suspect 3. They've got that top bar is the same, the 2 in the middle are the same, and then that last one is the same. So to me, suspect 3 and the crime scene have the same DNA fingerprint. So that means that suspect 3 is most likely the person who left this DNA at the crime scene. So let's go with suspect 3. Okay, and then it also asks why. How do we know that? So I'm going to put the DNA at the crime scene. And let's put matched suspect 3. Okay, so the DNA at the crime scene and suspect 3 have the same DNA fingerprint. Okay, matched number 3. Great, so number 3 is probably the person who did this crime. Let's look at the next one. Number 2 says, in a strange turn of events, one of the suspects turns out to be related to the victim. Who most likely is related to the victim? Okay, so for this one, where the first time we were comparing the bands and we wanted to find a direct match, because these 2 individuals are just related, they probably won't have exactly the same bands. Unless they're identical twins, they're not going to have the same DNA. So what we really want to do here is look at the victim's DNA fingerprint and compare them to the suspects and see which 2 are the most similar. They should have at least one band in common, and that's most likely going to be our relative. So let's see what we got here. Okay, suspect 1, not really matching at all. Okay, suspect 2 actually has 2 of the bands in common. So you can see this one is the same, this one is the same, but then suspect 2 has an extra one here. Let's see if anyone else is matching up any better than that. Okay, so this individual here, okay, they've got 0 bands in common with the victim, and then suspect 4 has 1 band in common. So overall, the victim and suspect 2 had the most bands in common. They had 2 bands in common. So that means that suspect 2 and the victim are probably related somehow because they share a lot of the same DNA. So for this one, let's put number 2. Okay. Great, so that was just the first one. I've got another one at the bottom. I hope this is kind of making sense. Again, what we're doing here is when we're trying to find an individual, we just want the known DNA to match up as well as we can with the individuals who may have done it or may be the father. And then if it's just talking about being related, they might share a couple of bands in common, but all of their DNA is not going to be the exact same. So let's look at the next one. Okay. So this one says, you are a scientist studying whales. You have recently helped a mother give birth to a calf. It is your job to find the most likely father of the calf. Use the DNA fingerprints below. Okay, so for this one, a little bit different. We have the calf's DNA and we have the mother's DNA. Okay, well, we know that half of your DNA comes from mom and half of your DNA comes from dad. So let's look at the calf's and the mother's DNA side by side. So we can see that these two match, right? The calf has this band and the mother has this band. There are 3 bands that the calf has that the mother does not have. So what we want to do here, these 3 bands must have come from the father, right? Because half comes from mom, half comes from dad. So one of these 4 males should have these 3 bands in common with the calf. If they do, that's going to be the father. Okay, so let's compare and contrast and see who the likely father of this calf is. Okay, male 1 does not have anything in common with the calf, so this is not the father. Let's look at male 2. It's got 1 band in common with the calf. So let's put a question mark by that one. That one is sharing a little bit, but not quite as much as we need. Let's look at male 3. Male 3, the same thing. They've got 1 band in common with the calf. So I'll put another question mark by that one. Okay, let's look at male 4. So they've got this band in common with the calf, and they have the 2 at the bottom in common with the calf. So that means that male 4 is going to be the father. And we'll put they share the most DNA. So again, when you're trying to find parentage, like who the father is of an individual, they're not going to share all of the bands together because the dad and the calf are not genetically identical. But because we knew this one shared, the calf got this band from the mother, that means that they must have gotten these 3 from the father. And only male 4 had those 3 bands. So that means that this is the mother and this is the father. Okay, so let's look at number 4. This says, after your research, you determined that one of the males must be related to the mother. Which male is the brother of the mother? So again, here, what we want to do is look at the males and compare it to the mother's DNA, and they'll probably share a couple bands in common. Whichever one shares the most bands in common with the mom is probably the brother. So let's compare and contrast. All right, male 1 has 2 bands in common with the mom. Okay, that's pretty good. That's a good chance that this is going to be our brother. But let's just compare it with the other ones to see if there's any more in common. Okay, male 2 does not share any bands with the mother, so this is not the brother. Okay, male 3, same thing, nothing in common with the mother. And then finally, we know that this is probably the dad, so hopefully this one is not related to the mother. They do not share any DNA together, so that means through this whole process, male 1 is probably related to the mother. So we got mom, we got dad, and then this is the uncle of the calf. So I hope that made sense to y'all. Y'all will get some more practice with this online. Thank y'all so much for hanging in there with me today. Make sure to give those other assignments your best effort, and I hope everybody has a great day.